So we'll start with a quick overview of today's agenda. First, we'll share a little bit of background on the ATMP Visible Particles team and why they formed and the problem they were trying to solve. And then we'll move on to the key messages of the publication. So we'll talk a little bit about the unique characteristics of cell therapies and the sort of challenges associated with those unique characteristics. We'll discuss the regulatory hurdles that many manufacturers face and how to assess the risk of patients' safety when it comes to the particulates. We'll also touch on the difficulties associated with visible inspection challenges and considerations around that in this space. And of course, there will be time at the end then for Q&A from the team. During the webinar, feel free to drop your questions into the chat, and we will have time at the end to address any of those questions. So it'd be great if we had a few questions from the get-go to ask at the start. So as you start to think of questions, don't wait for the end, drop them into chat and we'll pick them up. Okay, before we do dive into the technical information, I do want to talk a little bit about BioPhorum, just because I appreciate if some people here aren't familiar with us and who we are. So BioPhorum really is a global collaboration platform that brings together the biopharmaceutical and medical device industries. We currently have over 150 member companies with over 7,500 SMEs that are working together and collaborating in this space. And our mission really is to create an environment where members can connect, collaborate, and accelerate progress, and ultimately driving industry-wide change. In the past year alone, we've had over 170 publications, presentations, and resources. And we're currently running over 200 global programs from Change. In terms of what makes BioPhorum unique, we're member-led, we're safe and confidential, and we focus on technical discussions. And this allows for open sharing of challenges and solutions. And ultimately, we're hoping it leads to cheaper, faster, and better journeys for all those involved. And the Visible Particles team, who you're going to hear from today, it's a great example of this collaborative spirit. So we brought together experts from the industry to tackle a very complex and I suppose key challenge for cell therapy manufacturers. You're going to hear a little bit from them today. So the team did consist of, it was actually over 15 member companies. The member companies here today are those who are able to provide approval for these slides. But if you look at the original publication, you will see the list of the full companies involved in the publication. So some key players there in the industry. You'll be glad to hear that's all from me for now. So I'm now going to pass it over to one of our key contributors from the team, who is going to talk a little bit about the publication and some of the highlights and key points. And I think, Joseph, you're going to take control. Is that right?
Yep, I've got control right now.
Oh, you've got it ready. Brilliant. Okay.
All right, great. Thanks so much for the introduction. My name is Joseph Wood. I'm currently a senior director at Regeneron in their Global Process Sciences group. I'm overseeing the advanced therapy labs as well as providing some MSAT support to our Regeneron Cell Medicine over in Cambridge. Prior to this, I was working at Bristol Myers and just Celgene and Juno Therapeutics on cell therapy products, and spent the better part of a decade at Genentech prior to that, working on sort of traditional biologics and practicals. So before getting into it, I do want to acknowledge the entire team. I'm just presenting. There was a number of people shown on the slides before, got legal approval beforehand, and there's even more that were part of this group. And all of us rely on our colleagues internally and throughout the industry for all the feedback gained to inform our perspective and put this presentation and publication together. So thank you so much for that. So the position of the publication, the problem, again, was the unique challenges that cell therapies present in terms of controlling them and detecting them compared to other particles. One big issue is that sterile filtration is not feasible. You'll filter out your cell therapy product if you make an attempt to do so. The small batch volumes, especially with autologous cell therapy, make it so that sampling destructive testing also not feasible. The background itself contains particles. It can be opaque. The containers can have difficulties, making it difficult. And then the health authorities are expecting us to sort of comply with the existing regulations that are out there. But these regulations are evolving with the industry. And our publication seeks to sort of tackle that, take a look at the current regulations and what others have done, and try to piece together a holistic strategy to address these challenges. So in this presentation, just because our publication is sort of hot off the press, I'm going to be hopping back and forth at key spots just to show you some additional content that's now available in our publication. Here is a QR code. I'll give you a moment to scan that. It'll provide a link to the publication. Just to kind of give you a quick introduction, though, you'll see the content is rich with a review of the current guidance, different challenges and limitations. We look at things like patient safety impact. When is it appropriate to consider a risk-based approach to release, even in the event of finding a particle? Root cause and characterization, control strategy. And then my favorite part is the appendices, because these are actionable checklists for internal audits or setting up your own visible particle control strategy. We got a summary of guidance that we reviewed, taking a look at some warning letters, how others were held accountable for some issues around visible particles, and then the survey results among our team that helped drove these initial conversations that landed us to this paper. There's about a dozen different figures in here to kind of illustrate our processes and concepts introduced, as well as about a half dozen different tables, which I'll present a handful of these today. So overall, we're gonna kind of go through just the flavor of the publication. It's split up into 5 different parts. One, overviewing the unique characteristics of cell therapies, the regulatory challenges, how they're evolving and don't necessarily fit us to start with. So we need to adapt. Visual inspection challenges due to the matrix and containers. Potential impact of particles. So when we do find a particle, how do we take a risk-based approach and judge it against the therapeutic benefits and make sure that that patient gets reliable medicine on time. One thing I love about the publication, the whole thing is very patient-centered. It's sort of not taking the easy way out, instead taking a very nuanced, detailed approach at assessing risk and making sure that patients can reliably get quality medicines. And then lastly, particle control. There is a large collection and a life cycle approach to controlling particulates, starting at early pre-IND process development, where you're starting to build your library, implement various manufacturing materials, quality, et cetera, controls here to reduce the occurrence and improve detection. The publication, as I mentioned before, has a number of different figures. We'll show these in greater detail later on in the presentation, but there's a very useful decision tree to follow. These are the starting point for when you find a particle. And this diagram just sort of showing that we really emphasize a life cycle approach and the importance of characterization of particles, whether they're inherent, intrinsic, or extrinsic in nature. Just to kind of align on definitions quickly, we've got extrinsic particles, which are foreign and unexpected particles related to the environment, equipment, packaging, or personnel. Things like cellulose and plastic, which were, according to our survey, some of the more common particulates found. We got insect parts, plastics, rubber, etc. That can be extrinsic particles. There's an expectation that when you find extrinsic particles, they are rejected. Intrinsic particles, this is where a more risk-based approach may be appropriate. These are particles related to the formulation ingredients, manufacturing process, or packaging, may form over time due to instability of the product or due to interactions between the product and container surfaces. So things like silicone oil, aggregated protein, rubber, plastic, glass, etc. And then this is sort of the hallmark of cell therapy, the inherent particles. These are the particle types that's expected to be present. So when you're developing your process, it's essential to know what the end product looked like. Start characterizing just even qualitatively what your aggregate cells may look like in your drug product. By the time, even post-thaw when they get to the clinic, resuspensions may be part of administrative or administration, et cetera. So there's all different forms once you get your final product into your final product container, the vial or the bag, and that is dynamic throughout the life of the drug product. So characterizing what that looks like and measuring it with each process change or each implemented change with potential impact to inherent particles. So those are our three big buckets of definitions for particles: extrinsic, intrinsic, and inherent. And we'll go over some ways to characterize these particles and these different classes of particles. And they all sort of feed into the orange here, which is your overall control strategy. So unique characteristics of cell therapies. This is our first table in the publication. It has, it looks at the bulk size or the lot size, the product lot size, contributing manufacturing steps, most significant particle control step, and the sampling and acceptance conditions for autologous cell therapy, allogenic cell therapy, and then typical parenterals for comparison. So it's one patient, one lot, or two lots, depending on the process for autologous cell therapy. But for allogenic, the idea is you manufacture it in bulk and deploy it to the market, similar to parenterals. The product size lot, you will be dealing with small lot sizes. In our survey, typical lot sizes for autologous were around 10 vials or less. Allogenic, the lot size can vary, and parenterals, you're typically dealing with much larger lot sizes. The manufacturing steps, this is a piece that, again, I mentioned the sterile filtration is a key consideration here for parenterals. There's a sterile filtration step prior to the drug product vial filling. For us, you can't have the cell therapy process is you can't have that sterile filter. One alternative control that we highly recommend in this paper, and not everyone is using it, is a 40 micron plus or minus filter or strainer, something to catch visible particles. I'm going to mention this again just because I think it's one of the most actionable parts of process development that can help you control for particles. One is if there is an instance of a particle, the scope of your investigation gets limited, at least in a risk-based way, to look first at filter and downstream of the filter. It also helps your material control strategy, where you can, once you know that there is a capture step, the consumables used downstream of that capture step, the filter could, you can put some more emphasis with your supplier quality resources and programs, et cetera, to control for intrinsic particles from the process. So again, the sample size is smaller. So with allogenic and parenterals, you can use AQL sampling and take that subset. But with the allogenic samples, yeah, so I see questions popping up in the chat. We're going to have Q&A at the end, but thank you for putting them there. Continue to please put the questions there. We have 2 times 100% visual inspection for autologous cell therapy. Both autologous and allogenic cell therapies can be considered difficult to inspect products, or parenterals vary. So we can't take a one-size-fits-all approach with the regulatory guidance out there. We kind of touched on that. We know that there's poor detectability in the cell therapy products because of the container as well as the matrix itself. The small batch sizes limit your ability to characterize and do destructive testing to inform your knowledge of the particles, both for the lot of interest as well as for the life cycle of the product. Another topic I haven't touched on yet is the short processing time. So I've been in places where we have to time the inspection of the entire batch working in allogenic cell therapy, just to make sure that we are not exceeding a time that could have potential impact to the product quality. I think the standard that people are sort of arriving at is around 5 seconds per vial for inspection in the visual inspection light booth, the black-white light booth, under around 3,000 lux. However, this has mechanical stresses, thermal stresses, time, and then light exposure, all to consider. So you have to have something efficient and thorough. The difficult to inspect products, again, so this is there's a time aspect of this too. So let's say you do find a particle and you want to take a risk-based approach. It's important to prospectively build up your knowledge base through characterization, because if you're reactively trying to characterize and come up with a risk-based approach, that timer is ticking and the patient is waiting for their medicine. So whereas if you're producing a large bulk of drug product, say in parenterals, there isn't a patient literally waiting for that lot release in most cases. For cell therapy, autologous, one patient, one lot, they're waiting at the clinic for you to make the decision. So regulatory challenges. The current guidance, we looked at the USPs, European Pharmacopoeia, Japanese Pharmacopoeia, FDA draft guidance, PDA tech reports. They make statements, some acknowledge cell therapies and ATMPs more explicitly than others. However, none of them give a complete guidance on how to go about visual inspection of particles in your visible particle control. And that's what this paper is aiming to inform. Sorry about that. I'll skip that. Apologies. So let's take a look. The USPs, 1788, 1790, and 790, they were developed around transparent, low viscosity, large batch size parenteral products. The supplemental destructive testing is required when the nature of the product or container limits visual inspection of the content. However, as mentioned before, we can't do destructive testing in some cases. The European Pharmacopoeia acknowledges cell therapy and ATMPs and says that these should fulfill the requirements of being essentially free of visible particles. This word essentially, kind of leads way for interpretation, and this paper seeks to kind of provide an interpretation, a risk-based approach. The FDA draft guidance suggests that foreign visible particulates, in other words, extrinsic particles, must be rejected. PDA Tech Report 79 requires both non-destructive and destructive testing and suggests a life cycle approach. There is more details about our review of these different regulatory guidances, looking at the key points of each, as well as the challenges that it presents to ATMBs and cell therapy specifically in the appendix of the publication. So if you're interested in reviewing that with your team, please go take a look. Next is visual inspection challenges. So they have the unique challenges compared to other modalities. We talked about that. And just to kind of show here some of the different categories, we have the manufacturing processes. Cells, they typically range between 5, 20, even upwards of 30 micron, which is just below the threshold for visible particles, typically considered around 50 to 100 microns. So when you get an aggregate of just a few cells, it starts to enter into the visible range. And what you'll see is these can aggregate into strands, etc., which can be confused with things like white threads or other white particles, or even block your ability to successfully detect particles floating in the matrix. There is dimethyl sulfoxide and cryoprotectants in most of these cell therapies because the next step after filling into the vial is cryopreservation. This chemical exposure can impact the product quality over time. So again, the clock is ticking to perform your visual inspection quickly. Difficult to inspect parenterals. The cell matrix can be categorized as DTI, difficult to inspect, again, because it's hazy. And then another piece I want to kind of spend more time on is operator qualification. So this is, at least my experience and others on the team, this is where you can really come up with a cell therapy specific type of strategy or operator qualification that may differ from your parenteral operator qualification process. Because you don't have the sterile filter, and visual inspection and detection of particles needs to be more holistic and looking at entry points to the process at a heightened level of risk versus just the drug product container. In your operator qualification, you may have kits or testing that reflects those single-use components that may be downstream of your 40 micron filter. If you don't have that, all of the consumables of interest or representative samples where you can put spiked particle standards or mock particles. I've seen putting marks with markers even and measuring them with image J to make sure that they're a certain micron size. And seeing that inspectors, operators with visual acuity testing can reliably detect certain particles over a 70% detection threshold is what's kind of the standard. So when you're developing your operator qualification, you create a kit that has the particle standards, the extrinsic particle standards that are, you're used to, the hair, the rubber, the glass, etc. But you may also include, consumables, etc., to detect for intrinsic particles, especially known intrinsic particles that are in your knowledge library, and have images of these as well as part of the qualification and training. This helps you get more detection points, which lowers your risk score. And also you're training them, this is a different aspect, not just the pre-use inspection of those consumables when they arrive prior to release in the manufacturing floor, but you can also qualify your operators to perform a point of use inspection. So while they're in the grade A environment in the BSC doing an open manipulation, for instance, for a filling process, you can run detection threshold studies with that lighting, typically on the order of a few hundred lux, with the gowning, everything like they are in the manufacturing environment, and test how reliably those operators can detect mock particles or particles, particle standards in consumables of interest, so that when they're actually running a GMP process, with a certain level of certainty, a measured detection threshold probability of detection, that they can reliably detect particles just at the point of use. And this adds another level of assurance to your control strategy. Stability, again, we touched on the dimethyl sulfoxide and cryoprotectant and light, etc., that could potentially be impacting things. One thing I do want to point out is that different regulatory guidance kind of is more, I would say the Japanese, for instance, the 10,000 lux requirement for difficult to inspect products, that should be measured, in my opinion, against the 3,000 lux or so recommendation in the USPs to see if you're actually getting an improvement on probability of detection, because you're adding more light to the process and more exposure. So it could potentially impact stability. So having studies to demonstrate that you have sort of optimized around lux without overbearing the light exposure is important. And then the containers as well. The material suppliers for these containers, it differs for cell therapies. When we surveyed the people on our work stream, I think all but one used vials only for their drug product filling. One member had bags, and on the commercial market, there's a combination of vials and bags. In both of these instances, you have plastics that could be, semi-opaque or have completely opaque portions to it. In the case of some vials, for instance, that could block your ability to see. And add an additional challenge to detecting particles. Labels as well should be mindfully placed. Chain of custody is extremely important in autologous cell therapy because it's one patient, one lot. So making sure that you are maintaining the chain of identity for the drug product container with your labeling, while at the same time not blocking the view of visible particles is important. Potential impact of particles. So this gets into a very actionable piece of the control strategy, things that point to when it may not be appropriate to consider disposition of the lot, depending on what the patient's needs are. there is this, here we go, diagram in the paper. We're not going to go through the entire flow chart, but it starts up here at the purple oval. That is a particle detected. And it goes through a number of decision points, seeing if things are extrinsic or intrinsic or inherent, checking to see if your lab testing is conclusive or not, queuing remanufacture, or in some cases, considering, can the dose be met if we hold on to this sample, or should we consider further assessment and lot disposition of the sample to make sure that the patient gets the product that they need? So this flow chart may look, simple, it's logical, but there's a number of points here, these blue squares, where the input of knowledge is essential for understanding what your risk state is, understanding, leachables, or if your particle could be filtered during administration, et cetera. There's all sorts of considerations at play here that could go into your internal procedures for lot disposition and analysis of particulates and the response to these. All of this will, over time, continue to build your knowledge base throughout the life cycle of the product. So I encourage you to open this up and take a look at it with your team. It's meant to be a starting point for discussion. Obviously, customize it for your drug and your company's policies and procedures and experience. So that is just sort of a starting point, how to assess risk to patient when you see a particle. So some other key questions in there is your product extensively characterized? Again, knowing what the final drug product is going to look like from batch to batch, and also knowing what these intrinsic particles could look like, having a good relationship with your suppliers, having clear visibility to what particles they know have a higher potential for entering your process or being sort of a material defect. These are all part of characterization. Is it possible to arrive at a point where destructive testing is not required? For destructive testing, yes, you get to isolate the particle and analyze it, but you've now lost X mil of drug product that could have gone to the patient. So asking that question throughout your decision tree is important. And then, essentially, at the end of this, is there a quality-based approach for release of particle-containing materials? This one, this is something that was definitely a paradigm change, going from biologics to cell therapy. You see a particle in a vial, usually one may be used to taking a more conservative approach because the knowledge gathering and assessment is not associated with such an urgent risk to the patient. So again, a very patient-centered approach here, asking how can we reliably get quality product to the patient is at the heart of this. So coming up with a quality-based approach that considers the patient as well as everything going on internally to the manufacturing site to make a decision. Then last, we have particle control. So they have a higher risk of occurrence because there's no sterile filtration, and we have to integrate it through multiple aspects. So this is sort of the holistic term in this paper, because we want people to not have one control or two control or even 5 controls they're relying on to prevent particulates, but at every opportunity, and there's a diagram we'll show that kind of goes over some of the controls, you have an opportunity to gain knowledge and information about your process, the inherent, the intrinsic particles especially. So we overwork in deviations. You're probably familiar with this. It's a Ishikawa diagram, sort of split up among the 5 M's: man, method, machinery, material, and environment or mother nature. This is a very helpful way to dissect and create a framework for understanding sources of particles. Each potential source of a particle is a potential place where you can put in a preventative action or a kappa or some sort of corrective action if something has occurred. Having this mapped out in advance as well helps guide investigations so that you can process them in a timely manner and get to disposition if the patient's waiting on the other end. So have a specialized, dissection of your environment, your sources of particulates throughout your process, ready and at hand and to be used during events. This is a Swiss cheese diagram. I've seen diagrams like this in layers of protection analysis from cross-contamination. In this instance, we're using it for particle control, where the first sort of slice of cheese here is process development. Again, is there a 40 micron filter in your process? Is there some other controls in your process? Is it closed every opportunity? Is it very limited open processing? These are all decisions that go in this first slice of cheese here. And I want to mention that this is something that could be looser potentially earlier in process development, but as you gain knowledge and experience with your particular product, it's expected that you sort of tighten the controls around your process. So there will be process improvements to avoid particulates if they're not already there by the time you get commercial. Next is supplier oversight. Having a good relationship with the suppliers, transparency of documentations, understanding what their defects look like industry-wide as they distribute them to multiple customers. Material selection. So how are you working with your supplier quality and procurement teams to sort of scrutinize the different options out there? Are you settling just because early in process development you used one thing and it would take extra effort to go find a different source that may have better particulate control? The expectation is that you over the life cycle of the program, make improvements to material selection if they're available. Incoming inspection. Prior to release of key consumables, such as the drug product files, et cetera, maybe doing AQL testing or point of use inspections, having them planned later in the process. Environmental and engineering controls to limit extrinsic particles from the environment. Process and material controls. This could be, those point of use inspections for the materials, as well as your materials management processes and distribution, logistics. Training, so I throw operator qualification in here. I sort of covered that earlier, but again, you can have very specialized, enhanced operator qualification, not just the focus on can I see a particle in the vial after it's already happened, but can I also see a particle on these consumables used, especially during open processing before they get a chance to enter. Drug product filtration, making sure you have filters if available or strainers. One thing the paper touches on is that even though it's not part of your manufacturing process, during administration, there could be filters or strainers in the line. If those are available or part of it, those do serve as an additional control, as well as instructions in the physician's insert. Although they're not as reliable because they're out of your control, they're in the clinic. Instructions for visual inspection just prior to delivery to the patient is another form of control. And then finally, when you do detect a particle by visual inspection, the trending and monitoring programs, some things slip through the cracks. For instance, I've seen at different companies, if you have a material defect with an intrinsic particle observed before use, that doesn't necessarily go into your deviation program. It may be a batch comment or something. However you're monitoring these instances, make sure that your trending and monitoring program collects all the information available to it so that it's not missing instances of particles found in consumables, especially. This could, because you could be getting a measurement that's not indicative of the true probability of occurrence of particulates. And so trending and monitoring programs should be dissected and be very kind of detailed and made sure to capture all instances of detected particles before and after use. So you may be asking what to do next. There is this beautiful checklist in the appendix. This is just one portion of it. It's multiple pages long. And you could use this to sort of audit your own program, build your own program. It's just, again, it's a starting point, but this is what the team came up with. So I'll thank you for your time. We've got a good amount of time for Q&A. It's 11:36, but I'll just end with, in conclusion, we got a proactive approach needed for pre-IND, so start characterizing particles and controlling for them, and even planning future improvements as soon as you can with the process. And if you've got a platform where you have multiple programs, incorporate those lessons learned with each iteration of the platform as well. It should be a continued cycle of characterizing. Sorry, just because I didn't touch on it. Another way of characterizing your consumables is by doing rinse studies. Some of the participants of this work stream even rinse their drug product containers prior to filling, which introduces other risks around microbial contamination as well as residual rinsate. But it is an option, and you could also do these studies on the consumables from suppliers to see, you get a statistically relevant number of samples measured where you're collecting rinsate and then filtering to identify particles. You can start to build a library of the expected intrinsic particles from consumables if they do arise. Some vendors may already have this available for their consumables. Again, phase appropriate is the way to go. There's all sorts of considerations when you're trying to get novel medicines to patients in a timely manner. And these process improvements, we know visual inspection, visible particles is just one of many considerations. So phase appropriate strategy is recommended. And then for cell therapies, just an acceptance, some particulates are inevitable. Destructive testing may not be feasible. So that knowledge library is very important. Everyone needs to be aware of it, know where it is, be trained on it, because it helps you respond quickly to these instances and ultimately get a reliable product supply to the patient. The proposed value of this work, we hope will help refine your strategies with managing visible particles. We hope that having an industry perspective will help advance your procedures and give you confidence, so that when you do have to defend or present your strategies, it's informed by a whole industry's worth of perspective, as well as a risk-based approach and your own knowledge library and strategy custom to your company. And we hope that this provides a nice place to align in the industry, especially when engaging regulators. So they're hearing these topics, hearing these ideas, not just for the first time when someone comes up with it, but it's a story that they've heard before and a story that they're expecting, and a story they're familiar with and understand, so that the conversation can go smoother. You can get to other topics. And so what we hope and believe is that this will result in an improvement in product quality, reduced risk for nonconformances, and regulatory delays. Kathleen, would you like to take over at this point and facilitate the Q&A?
Sure. Yes. Thanks very much, Joe. That was great. Yes, I suppose in terms of next steps for this publication, ultimately we want to do is get this proposed approach out and roll it out to industry, share with some external partners. Within BioPhorum, we meet frequently with some of the external partners within this list, and we'll be showcasing to them and getting their feedback. We're having the webinar here today, and so we'd always love your feedback as well and your thoughts on the proposed approach. And ultimately then we're rolling it out at conferences. So there's a number of conferences here that we presented at so far, both via oral presentations and posters, and we'll be continuing to do that as we move into next year and the new conference season. So we're just trying to get the word out there on this proposed approach. As Joseph said, alignment is key. So it's always great to hear to other people's thoughts and feedbacks. So thank you so much, everybody. I encourage you very much to download the paper. And I want to just take this moment to thank the team. You've been a great team to work with. And hopefully we will come again together soon with some more additional content.